looks like a lot of you got it. I think the Hartnell server was acting a little wacky earlier, and so you might have had some trouble downloading it if you just tried to download it today. But as long as at least one person in your group has it, you'll be fine. And to do today's exercise, in addition to the handout that you printed out, you'll need a packet here. And once I get finished with the pre-lab lecture, I'll pass out these packets. But in regards to the pre-lab lecture, this is the part of the protein lecture that I skipped in the lecture hall, and I want to finish it off here. So get out your protein lecture notes and take lots of notes. Okay, well, one of the things I've been showing you a lot so far while talking about proteins is a little animation like you see up here, that proteins are one of the macromolecules, one of the 4 macromolecules along with carbohydrates and lipids and nucleic acids. But each of the macromolecules is built by linking together monomers, smaller building block molecules. And each of the macromolecule types has its own particular monomer. For proteins, the monomers are called amino acids, or to put that a little more simply, a protein is just a bunch of amino acids all linked together. And up to this point, I've just been showing the amino acids as little skittles, all linked together like that. But amino acids and all the monomers are actually just smaller organic molecules. And so what I want to do now is take a little bit closer look at the amino acids and see what they actually are at a molecular level. So to start off with, there are 20 different varieties of amino acids, and that's why I've been showing them as 20 different colored little balls there. But to see what they actually look like, let's imagine taking 3 of those amino acids. Alanine is one of the 20 amino acids, and so is valine, and so is cysteine. And kind of peel back the curtain for a minute and see what they actually look like. Here's what they are. You can see they're not too huge. Most monomers are fairly small. And for right now, ignore the blue parts. What I want to focus in on is the non-blue parts. If you look at the non-blue parts, notice they're all the same. And that's not just true for these 3 amino acids. All 20 amino acids have a part that's exactly the same. They call that the backbone of an amino acid, is the part that's always the same from one amino acid to another. And let me show you kind of a close-up of it here. So all amino acids have this structure as their backbone, and it has 3 major parts. The first part here is called the amine part. It's just named after the functional group. That is, an amine functional group is a nitrogen with some hydrogens attached to it. The second part of the amino acid backbone is this carbon right here. It's just called the central carbon. It's kind of in the middle. And the third part is this carboxylic acid functional group. And remember, a carboxylic acid is a carbon that has a double bonded oxygen on one side and an oxygen and a hydrogen on the other side. Okay, so that's an amino acid backbone. They've all got that. How do they differ? Well, here's how they differ. In all amino acids, there's another group of atoms that are attached here to the bottom side of the central carbon. And they call that the side chain, or the R group of the amino acid, and that's the part that's different. There are 20 different R groups, which is why there are 20 different amino acids. Let's see if I can show you an R here. Oh, I guess not. So I'm going to show you some examples of R groups here. And you do not have to memorize these specific R groups. As long as you know the backbone structure and that there are some differing atoms here that are called the R group, then that's all I want you to know. But just so you can see a few examples. The amino acid that has this particular R group is called aspartate amino acid. And for each of the 20 amino acids, it has its own name, and it also has a 3-letter abbreviation for its name, typically just the first 3 letters. ASP is aspartate. I guess I mentioned that here. Now, if you look at the R group of aspartate amino acid, notice that it is an ion. That oxygen has a little negative charge there. That's true. Certain types of amino acids have ionic R groups, like aspartame. Here's another amino acid. Let's get rid of that R group. And see this one. This amino acid is called serine. Serine amino acid always has this for its R group. And notice it's not an ionic R group, but it is a what R group? Not ionic, but polar. Yeah, I'm not drawing them in here, but you know that the oxygen will have a partial negative charge, this hydrogen will have a partial positive charge, this carbon has a partial positive charge, and so some R groups are polar. And this also illustrates the naming convention. Here's the full name, serine, or ser is it's the shorthand for this particular amino acid. Here's another amino acid. This one's called valine, or VAL, Val for short, and its R group looks like this, which you can see is nonpolar. Just all carbons and hydrogens, no partial charges. Nonpolar R groups are fine also. All right, so I'm just going to show them as an R. And I don't know why they picked the letter R to stand for the different atoms that you find there in the amino acids, but for whatever reason, that's the official letter that they picked. Okay, now a protein is a bunch of amino acids linked together, and so now let's see exactly how these amino acids get linked up to each other. 20 different amino acids, because there's 20 different R groups. Here we go. All right, so we're going to see how they connect onto each other. And let's start off, imagine we're going to start off with this amino acid. That'll be our first amino acid. Here it is. When a cell wants to build an amino acid, when a cell wants to, not build amino acids, build a protein by linking amino acids together, it always starts off with the first amino acid like this. The next amino acid that comes in always comes in this direction and brings its amine group, its nitrogen, inward like this, kind of backs the nitrogen up close to the OH of the carboxylic acid carbon like this. This is always the direction that the incoming amino acid comes in from. All right, so once the next amino acid comes in, what happens is this oxygen, this hydrogen, and this hydrogen get broken off, forming a water. And so this is an example of a what? Dehydration synthesis. Yeah, it's a little odd because most dehydration syntheses we saw are two hydroxyl groups coming together and losing a water molecule. Well, we're still losing the water molecule, so it's still dehydration, but now it's not exactly two hydroxyl groups. Anyway, once the OH has been removed from that amino acid and the hydrogen has been removed from the incoming amino acid's amine group, now the new amino acid can click into the carbon of the previous amino acid, click like that, and now we've got the two amino acids linked together by this covalent bond joining the carbon of the first amino acid to the amine nitrogen of the amino acid that just clicked in. And there's a special name for that linkage. It's called a peptide bond. I'm not exactly sure where they get the term from, but the peptide bond means the covalent bond that joins amino acids together when you're polymerizing them. And it not only actually includes the covalent bond between the carbon of the first amino acid and the nitrogen of the next amino acid, but it also includes the double bonded oxygen on that carbon and the hydrogen that was stuck onto the nitrogen. The whole kit and caboodle there is considered part of the peptide bond. All right, so now we have two amino acids linked together, but proteins are typically a lot bigger than that. I guess the average protein is 400 amino acids. But notice that we still have, now we have another carboxylic acid group over here, and so now we can bring in amino acid number 3 and connect onto that one. Let's scooch it over a little bit. Here comes amino acid number 3. Remember, they always come in, they bring in their amine group towards the previous amino acid's carboxylic acid group, You get the dehydration synthesis. And the amine group slides over and clicks into the chain of amino acids. Click. Now you've got another peptide bond. And now we have 3 amino acids linked together. And we keep going. Now this amino acid with its carboxylic acid group is there. And we can bring in amino acid number 4. It will click in place. We're going to get yet another peptide bond forming. And now we have, what is that, 4 amino acids clicked together. And now the 4th amino acid has its carboxylic acid group hanging out there. Now we can bring in amino acid number 5. Yes?
How about the C with the R group in it?
Uh-huh.
How come it doesn't have like a hydrogen?
Why doesn't this one get involved?
Because there's 3 bonds. So
Oh, I see what you're saying. This carbon only has 3 bonds. I left out the hydrogen sticking on top. Yeah, it's observant, but I didn't add another hydrogen on top of the central carbon. But yeah, you're right, it's supposed to be there. Good call. Okay, and there's no theoretical end to this process. We could just keep linking amino acids on and amino acids on and amino acids on. Let's see. See if I can convince my computer to end itself here in a second. Finish. Go back. Okay, there we go. This is a diagram from your textbook that summarizes this whole business that I just showed you. So there's the peptide bond in green there. Okay, so here's a drill. How many amino acids long is this protein? 6. So that's easy because I put them in different colors. But if I didn't write them in different colors, hopefully you would have been able to figure it out because you would have said, okay, amine group, central carbon, carboxylic acid carbon is the first amino acid, and then amine group, central carbon, carboxylic acid carbon is the amino acid number 2, amino acid number 3. You should be able to work your way along the backbone and figure out how many amino acids it is. Or just count the R groups. I guess that would have worked also. So here's a slightly trickier question. So yeah, 6 amino acids. How many peptide bonds are there?